Wonderful.
Thank you very much for giving me this time, first of all. So yes, it was mentioned before, I'm gonna go an overview on what is the current situation of the family Pentatomidae with the recent results that have been going on. So first of all, present like the group, why it's important. We know like pentatomids are important plant pests with one of the most important plant pests currently with a worldwide distribution, Alimorphalis, which is basically eating all the crops around the world. But also pentatomids are important because they can act as biological control agents. There is a small group that are that feed on other invertebrates, mainly holometabolous insects, which is the Azobins. And then we cannot forget the fact that many species are consumed locally as food, providing a good source of proteins to the diet. So first of all, previous to the analysis, the family was already recovered as monophyletic using a small data set of, or a small number of species, and using both morphological and molecular markers. The family is characterized by having a set of synapomorphies like the Gonapophysis 8 and the first Ramilos, the presence of triangulin, the Gonapophysis 9 reduced fused to the Gonacoccyx 9 with the second Ramilos, and finally the spermathecal duct is dilated and its distal orifice is provided with a sclerotized rod within it in its side. So with the information we had previously, however, the internal relationships of the Pentatomidae were not solved. Some of the subfamilies appear to be polyphyletic or non-monophyletic in this total evidence analysis. However, nothing could be extracted from that, as the amount of taxa included in the analysis was pretty low in terms of the family Pentatomidae, as the aims of this study was the relationships within the superfamily. Also, other results obtained from other molecular markers, in this case, obtained from whole mitochondrial genomes, obtained pretty similar results to the ones obtained in 2008. However, also with a very small amount of taxa, which therefore could not lead us to any information. As we can see, the results were partial, however, highly congruent with the previous ones obtained. Some of the groups within the Pentatomidae, however, have we have obtained, or groups have obtained phylogenies of them. And as in the case of the Antisine and the Discocephaline, they both were recovered as monophyletic, and the internal relationships were recovered too. The same case happens to some of the groups within the subfamily Pentatomine, which is currently a dump group in the Pentatomids, however, the nominal subfamily. And this is the case of some of the Asian Aliini, the Euchistus group, or more recently, the Chlorocorini. However, in all the previous results obtained, suggested that Tyrtocorids were placed at the basal lineage, as some of these orders already mentioned. However, after from the results of the phylogeny obtained, Tyrtocorids were excluded from the family Pentatomidae, which was already hypothesized by several authors. And as we can see here in the amplified part of the phylogeny, we can see that clearly Tyrtocorids fell totally apart from the Pentatomids, and their status was renewed as a family by itself. And this is after examining the Tyrtocorids, this makes some morphological sense, as they have, in one case, absence of triangulin. The second valvifer has a distinct medium fusion line, and the male Phylotheca is relatively flexible, which is not the case in pentatomids. However, what made some others think that cryptochorids might fall into the pentatomids is the fact that the spermathecal duct is dilated and it has some kind of sclerotized rod within it, which in this case would be from a different origin. So after the results obtained in the phylogeny, including so far the most number of taxa, we could decide that the family is composed by 9 subfamilies or 9 classical subfamilies until what we consider subfamilies until date. The subfamily Cervaninae, which was questioned if it belonged to the family Floidae or not by previous authors, clearly was separated from the Floidae and fell into a early diverging lineage of the pentatomids. Also, if we compare the internal female genitalia of these two groups, we can see that they are clearly different. And in the case of the Cervaninae, they follow this general Pentatomidae structure organization. Additionally, in the analysis, we found that Rhynchocorines, which is currently a tribe within the Pentatominae, always appeared as one of these early diverging lineages within the Pentatomidae. And this, in this case, this was obtained in previous results also, and also some unpublished results based only on morphology that I could extract from my dissertation. Additionally, some curious cases, the neotropical groups Chlorocorini currently in the Pentatominae and the subfamilies Discocephaline and Edesine, all from the neotropical region. They kind of form a group which we really do not know yet the relationships among them, but they are always they always appear as early diverging in the tree of the Pentatomidae. And this was also congruent with the previous results obtained from research that came out previous to the general phylogeny. So after these groups were more or less stable and the tree resulted from that gave us some clearly insight, we happen to have a big bunch of stuff that we really do not know what's going on there. However, besides that, we can more or less get some things clear. However, the relationships of these groups are still not well supported. Therefore, some groups could be recovered as monophyletic. Some groups were clearly not monophyletic in the analysis. And then, as some curious cases, some of the subfamilies that are currently recognized were recovered as sister lineages of Pentatominae tribes, which we will go on further in the presentation. So in first case, we can say that Philocephalines and Azobins are monophyletic with the amount of taxa that we included in the analysis. But on the other side, Podopines were not recovered as monophyletic. These results were clearly were already stated by previous results, where Podopines did not cluster together in a monophyletic group. However, in this case, by including more data, we could clearly see this differentiation. However, we still have all these Pentatominae sorted out together and without any kind of sense or logic. But unluckily, with the tree obtained, we had no support to establish any information or any classification based on that. We are sure that Pentatominae is not monophyletic, as clearly the neotropical Chlorocorines, the Rhynchocorines, and then some tropical bugs that were placed in the Pentatominae clearly fall out of the rest of this group. On the other hand, Carpocorini, which is one of the biggest groups within the Pentatomidae, is composed by multiple lineages. We can find two main clusters, which one corresponds to the old world and the other one corresponds to the new world. On the other hand, the old world Carpocorini was found to be paraphyletic because the current tribes Agonostelini and Nailini were clustered within this tribe, which these tribes probably will need to be synonymized under the Carpocorini sensu stricto, while the New World Carpocorini probably needs to be redefined as a tribe by itself. On the other hand, we checked with the phylogeny that some of the characters that are currently used to determine tribes are actually not that stable or should be taken into account in a broader sense. This is the case of the tribe Nephagini, which this tribe is characterized by the specimens or the species being green when alive and green when dead. This might sound a bit absurd, but according to the results we obtained on the tree, all green species in life that remain green in death clustered together in a monophyletic lineage. However, we had some additional species or groups of species that clustered together in this case. This is a case of tribes Methidaini, which are very adapted to their environment as they are leaf or Poathia feeders, some Antestini and some Pentatomini and some other place genera that fell inside this cluster. On the other hand, Menidini was found to be the sister lineage of the Azobins, and that obviously made at least me think if Azobine is currently one of the subfamilies within Pentatomidae, it cannot be the sister lineage of Menidini, which is currently a tribe within the Pentatomidae. And after examining specimens and checking the different parts, as Azobines had a very clear synapomorphism due to their rostrum specifically designed for feeding on other arthropods. However, checking on the genitalia, we could find that Menidini also possess some of the structures that are not unique to Azobines, but that all Azobines possess, that are the pseudoclaspers present on the male genital structure. This has led to find papers that tend to use the Azobines as outgroup of the pentatomids or of other groups, leading to wrong phylogenetic interpretations, and therefore leading also to possible wrong classifications within this new classification that we are trying to work with the Pentatomidae. So until date, what we have found extracting the morphology from the tree, even though there's not total evidence tree presented yet, we have seen that ventral thoracic structures had not been considered as important in characterizing groups within Pentatomidae, as most of these basal groups, or early diverging groups, better said, all possess some kind of ventral thoracic structure modification within most of the species. Additionally, genital characters that have been considered, but only in terms of smaller groups, were not interpreted in a whole of the Pentatomidae, and therefore they should be interpreted in the future in order to clear which groups share this set of characters, and showing that some characters should be reinterpreted in a way different to what we are doing until now, like, for example, the rostrum in Azobine. So for what we have now, the conception of what we have now of Pentatomidae, which we have clear subfamilies and clear tribes, maybe we don't have that much clear information, and we should start thinking that maybe what we thought that they were subfamilies, they might be tribes. What we had as tribes might be subfamilies, and what we thought they were tribes, they're just a bunch of species that group together that have some kind of morphological similarity. So I'm sorry not to have provided you with a whole new classification of the pentatomids, if that was what you were expecting. And luckily, we have still much more analysis to do using combined data sets, also with new approaches, UCs, morphology, in order to produce new phylogenetic studies. Obviously, new taxonomic revisions are needed because some of the groups are complete chaos, and therefore new species, genome revisions, and descriptions. And then all these will hopefully sooner than expected will happen to deliver us some new systematic accounts. But for all these, we will need huge international cooperation, as family Pentatomidae is one of the largest groups, and still with the previous results obtained, one of the major issues we had was the taxon bias, or the lack of samples from certain groups and certain areas. So still, as you have seen with the presentation, there is still loads of work to do. And I put this picture here of Astridotarcine, which some of you might have seen, I hope so, because we really do not know where does this fella fall, and it's currently one of the subfamilies within the pentatomids. And if you happen to have any specimen that we could extract some DNA from it, that would be extremely helpful for a future analysis, and also to know where this fellow falls in the tree. So thank you very much for your attention. I hope that I created even more questions than before with this presentation. That was a bit of the aim, to create some mental instability in how the internal organization of the pentatomids is. And please, I'll answer any question. And also, I would like to say, just to end up the presentation, remember that the next DHS meeting is occurring in Barcelona from the 4th to the 8th of July, hosted by Dr. Marta Goula and me. We have everything set up, everything's in place. We are just finishing and ultimating the last details. And also remember, all of you, that there's 2 workshops going on during the Congress. One is the workshop on Guerramorpha, led by Dr. Felipe Moreira and Dr. Lana Chen. And then there's second workshop on Pentatomoidea, which I'm trying to lead together with some of the colleagues, in order to basically try to understand what is going on with the Pentatomoidea, and more specifically with the Pentatomidae. And thank you very much for your attention.